Okay, hello. One of the sort of most important advances in psychology, particularly the last couple of decades, has been this ability to peer into the brain and see the neural activity that occurs during different behaviors. Now, researchers have a number of different tools in their toolbox to do this, all with their own relative pros and cons. So let's have a look at the different approaches to studying the brain. Specifically, we're going to look at electroencephalography, positron emission tomography, and functional magnetic resonance imaging. So, electroencephalography, or as we'll refer to it, EEG, is based on recording electrical brain activity at the scalp. So patients or participants would wear a cap that has a number of electrodes inside it that make contact with the participant's scalp. And it makes contact by, you put these little electrodes in, and there's a conductive gel that you apply in between the scalp and the electrode. And when neurons fire, they generate an electrical potential, and it is this electrical potential that EEG electrodes are recording. Now, if you think about the electrical potential that comes from a single neuron, this is way too small to be detected by EEG. But what EEG can detect is activity from multiple, i.e. thousands or even more than thousands of neurons that are all firing in the same region. So in a research setting, we're often interested in measuring event-related potentials from EEG recordings. And what I mean by that is that participants would be presented with some sort of stimulus. So you might present an object or a face on a computer screen, and then you record the electrical response that occurs whilst people are processing this stimulus that they're looking at. And doing this once isn't very useful a lot of the time, simply for the fact that you're recording this small electrical activity. There's a lot of noise or kind of randomness that's coming into the signal that you're trying to record. So researchers would typically present the same stimuli on multiple occasions and then average out the responses that they observe to create a kind of average waveform that indicates the electrical response for that participant. In this regard, evoked response potentials produce very precise temporal measures of brain activity. So they're accurate to within milliseconds. So you can know precisely when regions are responding to stimuli. However, one of the limitations of EEG is that the spatial resolution is poor. So it's hard to tell exactly which region is active. And we're generally limited to also only being able to assess activity in the cerebral cortex. So if you're measuring scalp activity, you can't really assess the activity of deeper brain regions. So for example, if you wanted to know what was going on in the amygdala, then measuring activity, which is deep down, measuring activity at the scalp, it's not really going to tell you much. Now, in terms of the spatial resolution as well, you can have different configurations of EEG electrodes. So you can have up to sort of 256 of these electrodes that might appear on the brain. And you can make some inferences that, yes, this kind of region is active, but it's limited to kind of sort of 5-10 centimeters you're active. So another method to study what's going on in the brain is using positron emission tomography. Now, this was primarily popular in the sort of 1980s and 90s. It's still used now, but maybe a little bit less rarely than it used to be. And it involves injecting radioactively labeled water. Not as dangerous as it sounds. And then after you've injected this radioactively labeled water, you measure with a scanning device the positrons that are emitted from it. So it works on the principle that when part of the cortex is activated, so neurons in that region are firing, the labeled water would move rapidly to that brain region, and researchers can then use this to infer the fact that you've got neural activity in that area of the brain. So a computer will use the data gathered by the scanners or the detectors to create some multi-dimensional, so like a 3D image that shows the distribution of this radio tracer in the brain. Now, one thing that positron emission tomography is useful for is the fact that you can use specific tracers or compounds that can bind to specific neurotransmitters to indicate neurotransmitter activity. So, for example, you can have a labeled form of glucose that will bind to specific neurotransmitters. One thing I would suggest right now is if you watch video 2.3, this will give you a great interactive overview of exactly how positron emission tomography works. Now, something that is very popular and that we'll cover multiple times throughout the course is functional magnetic resonance imaging. So, in other words, fMRI. And this is a neuroimaging technique that uses magnetic fields to construct a detailed representation, so a 3D image of levels of activity in various parts of the brain at any given time. Now, fMRI is based on some key principles. First is the idea that neural activity is systematically associated with changes in the relative concentration of oxygen in local blood supply. So in other words, when a region of the brain is activated, it leads to an increase in oxygenated blood that flows to these neurons. The second key principle is that oxygenated blood has different magnetic properties relative to deoxygenated blood. The third principle is the fact that changes in the ratio of oxygenated and deoxygenated blood can be inferred by fMRI. So you can actually measure these different levels by applying a magnetic field and then recording the level of oxygenated or deoxygenated blood. And again, there's 2 assignments for you to read that will give you a much deeper understanding of how fMRI works. So one of the strengths of fMRI is the fact that it tends to have very good spatial resolution. So in other words, you can understand exactly which precise regions of the brain are showing increased or decreased activation. So this can be as precise as 1 millimeter and lower. Now, some of the negative sides or disadvantage of using fMRI is the fact that it does have relatively moderate temporal resolution. So if you think about the inference that you're trying to make of brain activity, if you're measuring oxygenated blood, then there's around about a 2 to 3 second delay from a brain region being activated to when that oxygenated blood moves towards this region. And we're measuring that thing that happens 2 to 3 seconds after the activity has occurred. So it's very difficult to have a kind of precise understanding of the times that these responses are occurring. There are also a lot of limitations or restrictions to the types of tasks that can be performed. So if you're placing somebody into a scanner where ideally they have very restricted movement, then there's only kind of very basic cognitive tasks that you can give them to do. And generally the sort of responses that you would make are single or 2-finger button responses. So it's hard to study complex processes or particularly social interactions using fMRI. So fMRI and EEG are perhaps the kind of 2 approaches that we're going to encounter most frequently throughout this course and also for the rest of your degree. But they're not the only methods. Another one is magnetoencephalography, or MEG. And this is a technique that involves the measurement or relies on the measurement of magnetic fields produced by electrical activity in the brain. So at the cellular level, so looking at individual neurons, these have electrochemical properties that result in the flow of electrically charged ions through a cell. And electromagnetic fields are then generated by this slow ionic current flow. And while the magnitude of fields associated with an individual neuron is incredibly small to be negligible, the effect of multiple neurons, so if you look at sort of 50 or 100,000 neurons that are all excited together in a specific area, this can generate still a very small but a measurable magnetic field. And these neuromagnetic signals generated by the brain are a billionth of the strength of the Earth's magnetic field. They're that small. So a MEG scanner requires these superconducting sensors that can measure these very small magnetic fields. Now, obviously, to do this requires having a magnetically shielded room, so you're free of any kind of interference. So you also need to block out the background magnetic field from the Earth. It's also a very expensive setup in general, so there's only a handful of labs around the world that have these installed. And it's also the case that this can be a relatively aversive experience for participants. So if you ask people afterwards of their experience, a lot of participants will say that it wasn't particularly enjoyable. Okay, so here we can see a summary of the temporal and spatial properties of these three key neuroscience techniques that we've just discussed. Now, considering the temporal and spatial properties is important because our research questions that we're asking will often require prioritizing one or the other. So, for example, if you want to know exactly what region of the brain is associated with the process, you're going to prioritize spatial resolution. Whereas at other times, we might want to determine precisely when the brain is responding to a stimuli. So generally you find using MEG and EEG techniques, these tend to have the best temporal resolution. Whereas if you want a better spatial resolution, then fMRI is much better suited to this. So in general, the types of brain imaging techniques that we've talked about provide useful information about the timing and the location of brain activity during performance of a wide range of tasks. And especially when this information is combined with behavioral data. So if you're studying both the processes that go on in the brain and the actual performance, so the accuracy and the speed or ability of somebody doing a task, combining that information together can be very useful. However, brain imaging techniques are not some purely magical solution for understanding how the brain works. So we've had neuroimaging techniques for at least the last 20 years. However, there's still a lot that we don't understand about how the brain works. So we can consider three specific reasons why this is the case that we've got difficulties. So first, there is just the sheer issue of complexity. So when we say that a brain region is active during a task, researchers might mean that it is more strongly activated during the task compared to its activation during a baseline measure. But it's often hard to know what baseline condition we should be using. So typically, when we're studying things, we would compare what's activity during the task versus activity during a resting state. But resting is rarely ever purely resting. So it's hard to have a measurement of the brain where you find a region that is completely inactive. We also tend to typically observe very small changes in activation. So for example, in most fMRI studies, performing a task will only lead to an increase in activation of a region that's around about 1 to 5% of an increase. You can think of it like this, that if you're measuring a region and say at baseline there are 10,000 neurons that are active or firing that region, a 1% increase would correspond to the fact that you then have 10,100 neurons that are activated. So you're looking at subtle changes that are occurring. And although the primary focus is the observation of areas that tend to increase activity, it's also the case that decreased activation can occur in some areas. And there's sometimes when this may be really important. So for example, if you look at the pattern of activation in the motor cortex when people move their fingers, if you were to move your right index finger, then what you would observe in the brain is increased activation in a corresponding area in the left motor hemisphere. Sorry, the motor cortex in the left hemisphere. And that's because the left side of the brain controls the right side of the body. There's a contralateral mapping that's there. However, the interesting thing is that you find that the corresponding region in the right hemisphere, so the region on this side that controls this hand, this actually shows a suppression effect, so reduced activity, which there's evidence to suggest that this is probably quite important for us being able to control our behavior. So there's some complex interplay of regions that might be active or deactivated. Secondly, it's also very difficult to draw accurate causal inferences. So if an area is active during a task, we can't really be certain that it's necessarily responsible for the task. So for example, if you were to imagine putting someone into an fMRI scanner and you ask them to do a complex reasoning task, and a particular region of the brain lights up or shows greater activity, could you then conclude with confidence that this region is related to reasoning? Have a think about whether that's the case. What other explanations might exist for why this region is activated? Okay, one reason is maybe this region that's just lit up reflects the anxiety or the worry of being in a really noisy scanner and then being given this complex task and trying to avoid looking stupid. So there's two possibilities here. It might be anxiety, the stress of the task, or it might genuinely reflect reasoning. Either way, drawing a direct causal inference definitely gets more complicated than you think it might be. And finally, most research is based on the idea of functional specification. So this is the assumption that specific areas are specialized for a particular activity. But things are almost always more complicated than this. So there's likely to be important networks or patterns of activation and connectivity that are going to play an important role. These are not impossible, but they're much harder to measure with imaging techniques.